Connaissance. Recherche. Sciences.
Et sciences mathématiques.
Théorie.
Physique.
Philosophie.
Conférence.
Archéologie.
Nature.
So I would like first to thank the organizer for inviting me, and in particular, considering the general topic of this meeting, to have included a presentation that is not at all in natura, but is rather a classical mouse experimental device. Even if I think that long-term memory is not really often studied in the mouse. So I will first make a brief recall. It's really classical textbook immunology about the B-cell response that starts with a proper encounter and activation by initiating a germinal center reaction. So a germinal center reaction starts by a very intense B-cell proliferation and the activation of DNA modification events that are mediated by an enzyme that is called AID, and which will be activation induced in deaminase, and which will be the basis of our mouse model. So this modification includes somatic mutation and isotype switch, and allows mutants that display a better affinity for the antigen to be selected by the antigen that is deposited on a very particular network of cells that is called follicular dendritic cells. So the germinal center reaction lasts for a few weeks, during which it feeds both the effector pool of short-lived and long-lived plasmocytes and the memory pool that, in the absence of other marker in the mouse, is mainly characterized by the IgG or the IgA surface phenotype. So, to illustrate the evolution with time of the germinal center B cell and the memory B cell, this is an old work of the group of Takemori, but several groups like Schlumschick or Tarlington have done the same in a typical hapten carrier response. Here it's NPCGG. So you see that the germinal cell, the number of cells drops rather drastically, the cells involved in the germinal center reaction, that are characterized either by two markers that are equivalent, either GL7 or PNA. And you see that after 3 months, there are only a few hundred cells that remains in the mouse. And you have also a slow decay of the memory B cells, again, in this case characterized by the IgG phenotype and by the capacity to bind the antigen. And again, after 3 months, you're left with a few thousand cells in a mouse spleen. And in fact, there are very little work performed after the 2 or 3 months that follow initial immune response. So the question we wanted to ask in our model is that is there a role of the antigen in the maintenance of the memory? Of course, the work of Klaus Rajewski have shown that you don't strictly need the antigen, but it doesn't prevent the antigen for having a specific role in specific type of memory. And there is another way to state the same question, is to ask for how long is the antigen deposited on the follicular dendritic cell network after an immunization. The second issue that has been controversial for many years is that do memory B cells go back to germinal center upon a recall response? And it is really the work of Berek and Milstein who propose that, let's say, affinity maturation is an iterative process where cycles of hypermutation of the immunoglobulin gene and selection are responsible for the increase in affinity of the B cell. But in fact, the recent work from the group of Randy Noel has shown that upon recall, in fact, you mainly generate plasmocyte and you don't mobilize cells to go back to the germinal center. Among other questions is that do you have other B cell subset other than the classical IgG positive B cells that are mobilized in a recall response. And the last question, and how long does memory last? Does it depend on the strength of the initial signal, and is it maintained by constant proliferation or simply by cells that have a very long lifespan? So the model we chose to make is a system where B cell engaged in an immune response will be simply marked and irreversibly marked by a fluorescent marker upon immunization. As an immunogen, we chose let's say, a rather classical immunogen, which is shipwreck blood cells, but with the idea that it would, could be closer to a pathogen than a classical NP, a classical hapten protein immunogen. And so we use a very classical system, which is the rosa LOXP-YP reporter line, in which the expression of the fluorescent dye, which is a yellow fluorescent protein, is prevented unless you have a sequence that is excised by the Cre recombinase enzyme. And we inserted the Cre enzyme at the AID locus, so activation-induced citidine deaminase, which is typically activated during the immune response. And the originality of this system is that the Cre enzyme is fused to the estrogen receptor, so it's sequestered in the cytoplasm as long as you do not feed the animal with tamoxifen. And when you give tamoxifen, the Cre enzyme is activated and thus it can excise the LOXP site and the cell becomes yellow. So by coordinating the injection of tamoxifen with the immunization, you can very timely mark precisely the cells that are engaged in an immune response. And the protocol that we follow is two consecutive immunization with chip red blood cell. We also use NPCGG as a control, and we also did control animal without immunization. And we have three stages of tamoxifen feeding, twice after the first immunization and one after the second one. And after that, we followed cells on the basis of the acquisition of the YP fluorescence for up to 1 year after the immunization. So it's really what we call long-term memory. So again, the control animals are only the 3 ingestion of tamoxifen without immunization. And the system is working quite efficiently because after the recall response and the last tamoxifen injection, you can see that when you look at germinal center B cells, so characterized as with a B cell specific marker B220 and with the PNA germinal center marker, you can see that 20% of them have acquired, and again it is irreversible, have acquired the YFP, a fluorescent marker. And you see these rather numerous cells have problem with that are labeled with YFP in the germinal center. And so you can come back months after if you're patient enough. This is only 2 months, but we did that up to 1 year. So you focus on cells that have acquired the YFP marker, and you can ask what is the phenotype, what in a mouse spleen section, what is the localization, and upon cell transfer, what is the function. So you see that you have quite a large number of cells that becomes labeled upon immunization, and that is progressively reduced with time, but still can be easily detected after 1 year. But what was also a surprise is that you have quite a constant number of cells that are labeled without any immunization. And again, we have a reasonable animal facility, so there is no obvious infection. So this is something that we want to study, and I will not be able to tell you more about that. Is that there is really an endogenous, spontaneous activation of cells. Again, it has to be AID mediated, and cells that have a very long lifespan because you find them after 1 year. So the first surprise in this model was to see that after 1 year, you still see those FACS profiles are gated on the cells that have acquired the YFP fluorescent labeling. So in these cells that have been labeled during the SRBC immunization, you see that you maintain a population that has the characteristic of germinal center B cells for up to 1 year after the immunization. So the absolute number decreases with time, but they are still present after 1 year. And if you look by confocal microscopy, so those are germinal center generated at the peak of the recall response, and you see that you can find clusters, so it's much more restricted structure, but it's clear germinal center, and by FACS analysis you can really check that the B cell in this structure are all the markers that are classically assigned to germinal center B cells. And if you match with the PNA marker, you see that you have these YFP label cells at all the time points studied that are located within germinal center. So we call that persistent or residual germinal center structure. And these persistent cells within germinal center are in close colocalization with follicular dendritic cells that are marked as CD35 is a specific FDC, follicular dendritic cell marker. So they are in close colocalization with FDC and with cells, CD4 cells that are most likely T helper follicular cells. So we didn't prove that the antigen was really present in this structure, but it's highly likely that, again, there is a colocalization with FDC and CD4 T helper cells involved a deposit of antigens that lasts for up to 1 year in this model. And again, using the Chi67 marker, which is a marker for cell proliferating cells, you can see that in this residual germinal center structure, 6 months after the secondary immunization, you can see YFP cells that are proliferating. So YFP are the light orange that YFP together with cells that are not proliferating within this germinal center structure for up to 6 months. So in contrast, when you look at an immunization performed with a classic, again, hapten carrier protein antigen, you generate germinal centers that last for a few weeks. But 4 months after the immunization, you find indeed memory B cell with the YFP labeling, but you don't find any of these cells cluster within germinal center-like structure, indicating most likely that the nature of the immunogen is responsible for generating this persistent activated structure. So focusing by FACS analysis on these YFP labeled cells, we notice that in fact only a minority of them are IgG positive, which is again the classical canonical marker for memory B cells, with a large proportion being IgM. And in both subsets, you can find cells that have we use both markers that differentiate the germinal center phenotype, and you can find that both IgM and IgG positive B cell exist either within with a germinal center phenotype or without a germinal center phenotype. And so if you tabulate the number of cells and their evolution with time, you see that surprisingly, the IgM subset is the most prominent, but in fact it is also the most prominent in the control animals. So again, control animals are animals that are fed with tamoxifen without immunization. Again, pointing that you have an activated IgM population that is generated permanently in the absence of any deliberate immunization. And if you determine the mutation status, because those are cells that have been engaged in a germinal center reaction and that have undergone somatic mutation of their immunoglobulin genes. So we wanted to determine the mutation level within each subset. And each of these points is one Ig sequence isolated from this various subset. And what is very striking is that within germinal center B cells, these are the IgG, these are the IgM, at 6 months after immunization, you're able to accumulate a very large number of mutations. So this is an average of 28 mutations in a sequence that is about the length of an immunoglobulin genes. So these are numbers that are frequently seen in immunoglobulin sequence in human, but are never reported in a classical hapten carrier response in the mouse. So we wanted to ask what is the function of this subset. So we have IgM, we have IgG, we have cells within germinal center, outside germinal center. So we made a recall response after transferring subsets that are sorted on the IgM or the IgG phenotype on the PNA plus or the PNA minus phenotype, transfer into an animal that have been pre-immunized several months before, boost the animal just 2 hours after the transfer, and 8 days after, ask what is the phenotype of the YAP cell, again, only focusing without a priori on the fluorescence, asking what is the phenotype and what is the localization, and what is the function, what is the specificity using an anti-SRBC ELISpot assay. So, when IgM positive cells are transferred, whether they have or they have not the germinal center PNA marker, they differentiate 8 days after transfer into new germinal center B cells. And during this differentiation, they are able, again, to reinitiate the germinal center reaction and to initiate isotype switch, since 1/3 of them become IgG. And when you do confocal microscopy, again, it is only 10 to the 4 cells that are transferred in a mouse. And it's really amazing that you can right away identify them clustered in germinal centers. There are a small one and a big one, small one here and a big one on this section. And you see that these YFP cells reinitiate a germinal center reaction. Whereas when you focus on IgG positive cells, you transfer them into pre-immunized host. They only differentiate into plasmocytes. They are not able to go back to the germinal center and to reinitiate a germinal center reaction. And when you test this plasmocyte for the specificity, they are mainly anti-SRBC specific, using a lysate of sheep red blood cells. And when you look in confocal microscopy, you see that this expanded population is mainly located in the red pulp and has a CD138 phenotype, which is a classical plasmocyte marker. And again, you don't see these cells within any germinal center structure within the B cell zone. So, as a summary, you generate 2 major populations after SRBC immunization, and 6 months after immunization, if you probe the function of these cells, you see that IgM positive B cells are able to give rise to a new germinal center response and to a small number of IgM secreting plasmacytes that I had no time to describe. Whereas the IgG population is only generating IgG secreting plasmacyte, and there is also a part of this population that does not differentiate and so is most likely involved in self-maintenance. So as a conclusion of this model, what we observe is that when you use a particulate antigen and not a soluble protein, you're able to generate in the mouse long-lasting germinal center structures that persist for, we observe them up to 8 months, but in fact after 1 year, I'm sure they are there because by fax we still find PNA plus cells. So again, this seems to be dependent upon the structure of the immunizing antigen. But that the B cell memory against sheep red blood cells is thus composed of 2 different layers, one layer in germinal center, one layer outside, but both of these layers are composed of both an IgM and an IgG component. And when you boost the animal, and whether it is by adoptive transfer or simply by re-immunizing the animal, you see that the IgG subset do not re-enter germinal center reaction and has an immediate effector function, whereas the IgM subset reinitiates the germinal center reaction that will, of course, give rise to IgG memory B cell in prevision of the next encounter of the antigen. And if you take the vocabulary of the T cells, you would say that the IgG subset has effector function, whereas the IgM subset would be much more like a central memory subset. So, of course, if the nature of the antigen matters, should one vaccinate with soluble of particulate antigen? I'm obviously not able to answer this question, but the only thing we would like to study in this model is in a system where you have this persisting germinal center structure, what do they provide in terms of quality or quantity of the immune response? Are they able to perform ongoing recruitment of naive B cells and so constantly mobilizing new B cell clones? Are they able to feed the memory B cell pool, both in terms of plasmacyte or in terms of memory B cells? But is there a dark side of it? That is, that when you think of the very unusual load of mutations that we saw on the IgG subset within germinal center, is this population at risk for oncogenic transformation? And when we do sequencing, we see that there is clearly major clonal expansion going on in this long-lasting germinal center structure. And the last picture I would like to show is our section from human spleen, because it is well known that in human you don't find many germinal center in the adult spleen. So we labeled, this is, let's say, splenic sample. So this is a young child, and this is splenectomies that are performed, surgical splenectomies that are performed due to a red cell disease. And of course, these children are classically immunized before the splenectomy. And so you can see this ring, the marker is IgD, and the red marker is a marker of FDC in human. So you see the germinal center structure in this children's spleen. But in the adult, even though And this is an accident, explained to me due to an accident, so that there's no deliberate immunization performed before splenectomy. And when what you see, in fact, using this FDC labeling, is that you have very small structure, and within this structure, we were able to find B cells with a germinal center phenotype. So you have structure that are really reminiscent of what I describe in the mouse system, that evokes persistent and residual germinal center reaction that either have been induced by the vaccination or by the infectious past of the individual. And I will stop on that and acknowledge. So most of the work in the mouse has been made by one postdoctoral student, which is Ismail Dogan, with the help of many people in the lab. And I would like to mention Marc Descatoires and Sandra Veller for this first slide on the human spleen, which is really a work that has started recently in the lab and that we want to develop. And of course, this has been done in Necker, in the INSERM unit. I'm co-directing with Jean-Claude Weil. Thank you.